Good afternoon, everyone. I'm thrilled to have the opportunity to introduce Paul Soto. Paul and I go way back to graduate school at Emory University, where we were graduate students in Jack McDowell's lab. Previously, Paul did his undergraduate work at the University of Florida with Tim Hackenberg. And after graduate school, Paul completed postdoctoral training in behavioral pharmacology at the National Institute on Drug Abuse and the Johns Hopkins University School of Medicine. In 2010, Paul received the B.F. Skinner New Investigator Award from Division 25 of the APA. One distinctive aspect of Paul is that he's like a Renaissance polymath. When he accepted the award, he gave a talk titled, "A Marriage of Behavior Analysis and Molecular Genetics." Paul is one of the few scientists I know that could preside over such a marriage. To switch metaphors a bit from marriage, Paul builds bridges relating behavior and neuroscience. JAB recently published a special issue on neuroscience, and the introduction noted that to increase the explanatory or causal traffic these bridges can bear, they should be anchored in multiple locations, both in behavior and in the brain. And that's what Paul does. He keeps us anchored in multiple locations, and then he relates them in conceptually systematic ways. He's asked fascinating questions about receptor systems supporting choice and behavioral economic decision-making, the physiological and behavioral impacts of early life exposure to stimulant medications, and the nature of cognitive decline associated with Alzheimer's disease using transgenic mice. Paul's published over 30 articles in a variety of journals, including high-impact, multisyllabic journals such as Neuropsychopharmacology and Psychopharmacology. He's received grant funding from the National Institutes of Health, and he's recently served as an associate editor for the Journal of the Experimental Analysis of Behavior. Currently, Paul is an assistant professor in the Department of Psychology at Louisiana State University. Not surprisingly, he teaches a course in learning and behavior, and a course on drugs, brain, and behavior. So we're very lucky to have him here today to construct yet another bridge relating genetically modified organisms and behavior. Please welcome Paul Soto.
Good afternoon, everyone. Thank you, Jesse, for that overly kind introduction, and I want to thank Todd for inviting me, and thanks for all of you who are attending. So I'm going to tell you about a few different projects that I've had the opportunity to work on that have utilized genetically modified mice. Maybe one day there will be other organisms that I can tell you about. But first I want to start by saying that I think that biological and genetic technologies have advanced to the point that although we can't know everything that is happening inside the behaving organism, we can know a lot about what is happening inside the behaving organism. And I think that these advances offer opportunities for fruitful research collaborations that we as behavior analysts should be taking advantage of. Okay, so I'm going to tell you about a few projects. Some work I did a little while ago looking at mice in which receptor for particular neurotransmitters have been eliminated to probe the role of those receptors. I'm going to tell you about some work I've been doing with my collaborator at Pennington Biomedical Research Center in Baton Rouge, looking at eliminating particular signaling molecules. And then I'll tell you about some work we're finishing up using a mouse model of Alzheimer's disease-related neuropathology. And then finally, I'll just mention some of the other exciting technologies that I haven't had the opportunity to work with yet, but they're just amazing, and hopefully you'll find them as exciting as I do. Okay, so knockout receptors, knockout of receptors refers to the ability to generate organisms that do not express a particular receptor of interest. So just a brief review for anybody who's not familiar with neurotransmitters and receptors. Neurotransmitters are molecules that are released from neurons, and those molecules then activate, bind to receptors that are proteins expressed in the membranes of other neurons. So you can see in the graphic there The blue icons here are the receptors, and the little balls here are the neurotransmitter molecules. When those neurotransmitter molecules bind to those receptors, they change the shape of the receptor, and that can have various kinds of effects. It might open a channel and allow the influx of charged ions, or it may initiate a second messenger signaling cascade. But ultimately, it has an effect on the neuron that expresses those receptors. So if you're interested in the role of a particular neurotransmitter signaling through a particular receptor, one, albeit a little bit crude way, is to take that receptor out and see how those animals differ from animals that still express the receptors. So how do you do this? So this handy graphic from the NIH illustrates the first part of this. So you can rely on your handy molecular biologist to alter embryonic stem cells so they can alter the DNA in these stem cells and disrupt a particular gene that codes for the receptor that you want to eliminate. So they make the gene non-functional. And then they inject those altered embryonic stem cells into a blastocyst, and they put that blastocyst into the uterus of a host female. And when she gives birth, she will give birth to what are called chimera pups. And these are pups that have a mix of tissues that are based on the altered cells as well as the original cells contained in the blastocyst. Then you can take these chimera mice, denoted by the gray mouse with the black stripes, and you can breed them with normal wild-type mice. And what you get when you breed them are heterozygous mice, that is, mice that have one functional copy of the gene and one non-functional copy of the gene, or more wild-type mice. And then in the next step, you can breed those heterozygous mice together, and you get 3 types of mice from that breeding. You get more wild types with 2 functional copies of the gene, some more heterozygous mice, and most importantly, homozygous knockouts that lack functional copies of the gene. So they have 2 disrupted genes, and they do not express whatever that gene codes for. So in this first project I'm going to tell you about, we were working with dopamine receptor knockout mice, and we were interested in the role of dopamine, like a million other researchers, because it's been implicated in a number of important disorders and conditions like compulsive eating, schizophrenia, ADHD, and drug abuse. And if you're not familiar, dopamine is a neurotransmitter that's released by dopaminergic neurons and activates dopamine receptors on neurons. This just illustrates the role of cocaine in dopamine's effects. So dopamine are the little orange balls here, and the white images are cocaine. And cocaine blocks the dopamine transporter, which picks up excess dopamine and takes it back into the neuron, thereby elevating dopamine. And that's thought to be one of the primary mechanisms underlying cocaine's effects, as well as other stimulant drugs like amphetamines. Now, there are 5 different dopamine receptor subtypes, and they are classified into 2 categories, D1-like and D2-like. And they are classified into these 2 categories based both on their structure and function. D1 and D5 receptors, when bound by dopamine, increase adenylyl cyclase levels. D2 receptors, D2, D3, and D4, decrease the levels of adenylyl cyclase. There's a little more to the story, which I think is summed up perfectly by this simple diagram, so I don't need to say any more about that. Now, we took mice that had been genetically engineered to lack either D2, D3, or D4. So we were focused on this D2-like receptor category. And we also worked with wild-type littermates as controls. So we're comparing performance of the wild types to the knockouts. And we exposed these mice to a range of fixed ratio values where completing the FR schedule produced delivery of food pellets from this food pellet dispenser here. And we exposed them to the range of FR schedules under both open economy and closed economy conditions. So in the open economy condition, the sessions were short and the number of reinforcers was capped and they were fed outside the session. In the closed economy condition, the sessions were long and all the food that they received within a 24-hour period was what they earned within the experimental session. And we analyzed the resulting data using the Hirsch and Silverberg exponential model of demand, which is a bit complicated looking, but relatively simple in its form. Basically, what it says is that the log of consumption, that is total reinforcers earned typically, declines as a function of the fixed ratio value in this case, denoted by C in the equation, with a couple parameter values. Q0, which is the consumption level at zero price, so what the animal would consume if there was no cost associated with consuming that reinforcer. And then the K parameter, which defines the span of the equation on the y-axis. And then most importantly, for our purposes, the alpha parameter, which defines how quickly the demand curve drops from Q0 down. So to better visualize the effect of alpha, you can see these two curves here. The curve to the left was generated using a larger alpha value than the one to the right. So the idea is that alpha is inversely related to the reinforcing value or effectiveness of the consequence that you have arranged. So here are some results. First off, you can see that as you increase the FR value, sorry, these are from an open economy condition, in all the mice, as you increase the FR value, the total number of reinforcers obtained decreases with increasing FR value. Now, in the D2 mice, the knockouts had demand curves that dropped more steeply than the wild types, and the same was true in the D4s, although the effect was bigger in the D2. So the alpha value was about twofold larger in the D2 knockouts compared to their wild type littermates, and was about 1.5-fold larger in the D4 knockouts compared to their wild type littermates. And in the closed economy condition, you can see again the same pattern occurs, right? As you increase the FR value, total number of reinforcers declines in a way that's predicted by the equation or described by the equation. But under the closed economy condition, the only difference that we found was in the D2 mice. So in that case, the alpha value for the knockouts was again about twofold larger in the D2 knockouts compared to the wild types. Now, we initially interpreted this difference as suggesting that food has higher reinforcing value in the wild types compared to the D2 knockouts. But one potential alternative explanation that we thought about was maybe these mice are more sensitive to extinction. So as they get to these high FR values, there are long periods without reinforcer delivery, and maybe their responding starts to extinguish during those times. So we exposed the mice to two types of extinction, which I'll explain in a second, and we analyzed the resulting response rates in the extinction sessions using the equation depicted there, where R represents response rate, T represents time, or actually session number in extinction, and K is a parameter that governs how quickly the curve declines. So K is a quantitative measure of the rate of extinction. So you can see a larger K means a faster decline in responding during extinction. So as I mentioned, we ran two types of extinction sessions. One that we called extinction without food because there were no food deliveries during the extinction sessions. There were actually no scheduled consequences. And what you can see there is that responding in extinction sessions decreases across the number of extinction sessions. However, there was no difference between the knockouts and wild types for D3 or D4, but there was a difference in the rate of extinction in the D2 knockouts compared to the wild types. But it was actually reverse of what we were worried about. The knockouts have slower rates of extinction than the wild types rather than what we were concerned about. We ran another version of extinction where we actually delivered food response independently. It was actually yoked to the timing of food deliveries in previous FR sessions that for each individual mice. Again, responding did decline in repeated extinction sessions. However, there's a lot of variability, as you can see, presumably due to adventitious reinforcement of responses and/or discriminative stimulus effects of food delivery occasioning further responding. There were no differences for the D3 and D4 knockouts compared to their wild types, but there was again a difference in the rate of extinction in the D2 knockouts compared to the wild types in the same direction. So extinction proceeded more rapidly in the wild types compared to the D2 knockouts. Okay, so all of that work, to sum it up with one sentence. We think that dopamine D2 receptors partially mediate the reinforcing effects of food. I say partially because clearly responding is reinforced by food presentation in these mice, but it just seems to have lower value. Now, some future directions that we are considering. One idea is to evaluate the generality of this effect. So, do D2 receptor knockout mice, are they less willing to work for other consequences, sucrose solution, water, etc.? We don't know that yet. Another direction that we're considering is looking at the particular brain regions and timing of knockout. And I'll talk a little bit about technologies that will allow us to do that. But you might want to know, because in our knockouts, the D2 receptor is knocked out everywhere in the body for the duration of the organism's lifetime. You might want to know what happens if you knock out D2 receptors in VTA or striatum, or only in adulthood, or only in adolescence, for example. Now, another project that I've been working on that I didn't put in the abstract, but I want to tell you about, is a collaborative project looking at, instead of knocking out the receptor for a signaling molecule, you're actually knocking out expression of the signaling molecule itself. And so this is a project that is in collaboration with Dr. Chris Morrison at Pennington Biomedical Research Center. And Chris is really interested in how animals choose between different macronutrient food sources. So what makes an animal choose a protein-rich source over a carbohydrate-rich source, for example? And he's interested in the role of this hormone called fibroblast growth factor 21, or FGF21 for short. So just to give you some background on this, FGF21 is a hormone that's synthesized in your liver, and it's important for energy balance as well as insulin sensitivity. And it acts on type 1C FGF receptors. These receptors are expressed all over the nervous system, though, including the brain. And before I started working with Chris, he had been doing a series of experiments where he provided access to different types of solutions in the mice's home cage. So some solutions might be a maltodextrin solution, a carbohydrate solution, or casein, a protein solution. When mice are maintained on a normal diet, meaning has a normal amount of protein, they show a strong preference for a carbohydrate solution over a protein-rich solution. But if you put them on a low-protein diet, they show a preference for a protein solution. That's probably not surprising. Here are some quantitative data just to back up that claim. You can see in wild-type mice, when they're maintained on a control diet, a diet that has a normal amount of protein, the black bars here, they consume more maltodextrin than they do casein. But if you put them on a low-protein diet, the red bars, you can see that now they consume much more casein than they consume maltodextrin. So protein restriction moves them to a preference for a protein-rich solution over a carbohydrate solution. If you do the same experiment in mice that do not express FGF21, you don't get that effect. Right? So these mice, FGF21 knockouts, when they're on a low-protein diet, still favor malto over casein, suggesting that FGF21 signaling is critical for driving protein preference. So Chris was interested in whether protein restriction would increase the reinforcing effectiveness of a protein solution, and if so, would that effect depend on FGF21 signaling? So we took C57 black, normal wild-type mice, and FGF21 knockout mice, and exposed them to a series of FR schedules. And the reinforcer in this situation was a dipper presentation of a 4% casein solution. So instead of food pellets, we were using these dippers. The casein solution is in this little black reservoir, and the arm lifts up, and then the mouse can lick up the little, I think it's 0.01 ml of these dippers. Okay, so When we varied the FR value in wild-type mice, either maintained on a normal protein diet or a low-protein diet, you see increases in total responses as you initially increase the FR and then decreases. But at the high FR values, you can see that mice maintained on a low-protein diet respond much more for casein than mice on a normal protein diet. And that translates to differences in their demand curves, not surprisingly. So the demand curve in the normal protein mice drops off more steeply than the demand curve in the low protein condition, suggesting higher reinforcing effectiveness of casein when your diet is low in protein. If you do the same thing in FGF21 knockout mice, you don't get a difference really in responding. There's a slight difference here, but when you do a model comparison test, now I'm nervous about talking about any statistical comparisons. I'll shrink down here, but if you do a model comparison test, these alpha values are not statistically different. Okay, so to conclude that, low protein diets increase the reinforcing value of casein solution, a protein solution, and that increase was dependent on FGF21 signaling. Some future directions. So one thing you might ask is, well, maybe protein-restricted mice will work harder for any kind of consumable reinforcer. And that's one thing we've started looking at, and our initial results suggest that's not the case. So they respond at equivalent rates when the reinforcer is malto, but at much higher rates when the reinforcer is casein. So it does seem to be a protein reinforcer specific effect. And then the next step that we want to take is there are actually mice in which the receptor for FGF21 is knocked out only in brain. So you can ask, is it FGF21 signaling in brain that is responsible for the effect rather than at some other target in the system? So those will be the next set of experiments that we'll move to. Okay, so the last project I want to tell you about, which we've been working on for several years now, is uses these Alzheimer's transgenic mice. So instead of disrupting the function of a gene, what you're doing is actually adding genes to the mouse genome. So before I tell you about our actual data, just briefly touch on some facts about Alzheimer's disease. It's responsible for substantial loss of life. There are a number of people, approximately 6 million people currently living with Alzheimer's disease, and that number is expected to more than double in the next 30 years. It, along with other dementias, is of significant economic burden. So estimates for this year are that it will cost about $300 billion, and that will more than triple in the next 30 years. Now, the diseases start to begin with early brain changes, potentially years or even decades before the initial symptoms. The initial symptoms most people are familiar with, complaints about short-term memory, some confusion, and unfortunately, one side note about this, the fact that the brain changes are thought to occur so long before the onset of symptoms will pose a difficulty even if a cure is developed one day, because unless we have a way to screen people and diagnose them, they will have some damage due to the disease progression before we actually are able to detect that they are suffering from the disease. Unfortunately, as the disease progresses, symptoms worsen. People begin to have difficulties with activities of daily life, like bathing and meal preparation and planning. And then eventually in the later stages, they lose the ability to communicate and recognize loved ones. So it's a devastating disease. Underlying those symptoms are two hallmark neuropathological features. So amyloid beta plaques, these buildup of amyloid beta extracellularly, and then the development of neurofibrillary tangles within the neurons. And both of these together are thought to contribute to neuronal dysfunction and death. Now, in studies that use mouse models of Alzheimer's, the typical behavioral preparation is the familiar water maze. If you're not familiar with how a water maze works, you put the animal in this big tub of water. There's a platform that's submerged just underneath the water. They swim around, they locate the platform, they can climb out of the water. If you keep the platform in the same location, they learn to go directly to the platform. But Alzheimer's mice have difficulty getting better in this task. They don't perform as well as normal mice. However, I'm preaching to the choir here, but I think we should be using operant procedures to evaluate these mouse models. There are a number of benefits that I think operant procedures have over maze procedures. I'm not going to go through the list, but for my purposes, operant procedures are going to be much more useful for screening potential pharmacotherapeutics. So it's much easier to evaluate a range of doses, which we will need to do if we're screening potential pharmacotherapeutics. It's much easier to do that with operant behavior than it is in a water maze procedure. It's also much easier to vary the procedural parameters because it's automated and we can program that. We can evaluate drug effects under a number of conditions, potentially within the same session, to evaluate the conditions under which beneficial drug effects occur. So we've been working with this particular mouse model called the APPswe mouse model. And so they have 2 genes from humans inserted into their genome. They have the Swedish variant of the amyloid precursor protein gene, which codes for, can you guess, amyloid precursor protein. And they also have a variant of the presenilin-1 gene. And so the ultimate outcome of having these 2 genes inserted into their genome is the development of beta-amyloid plaques. So you can see from one of Elena Savanenko's early papers here, the increase in beta-amyloid, very low or non-existent levels in 3-month-old mice, increase in amyloid beta at 7 months, and then a very large increase by 19 months of age. Give you sorry, I'm out of time with my timing here. To give you a visual on this, these are stains from brain, so the little white splotches are beta-amyloid deposits in brain, and so you can see low to moderate levels of beta-amyloid in the brains of 7-month-old mice, and then substantial beta-amyloid deposition in 19-month-old mice. So we've been looking at these mice in delayed matching to position tasks. We've also been looking at them in a 3-choice serial reaction time task. I'm not going to show you those data today. We're looking at male and female transgenics as well as non-transgenic littermate controls. So the way our delayed matching to position task works is at the start of a trial, one of the levers is randomly selected and inserted into the chamber. Once the mouse makes a press on the lever, it's retracted and a delay is initiated. The delay varies from trial to trial randomly and can be anywhere between 0.1 and 24 seconds. The first nose poke in the food cup after the delay has elapsed produces extension of the 2 levers. If the mouse presses the previously presented lever, food is delivered. If they press the other lever, they get a 10-second timeout. And we actually split our mice into 2 testing groups. One that we labeled the intermittent testing group, and one that we labeled a continuous testing group. And the reason this group is called the intermittent testing group is because they take breaks. So they're initially trained between 8 and 12 weeks. They reach final parameters, usually by 13 weeks of age, and then they continue daily testing until they were 28 weeks of age, and then they take a break. And they come back at 41 to 52, then they take another break. And the reason that we did this was a concern that daily testing might actually prevent the development of deficits on our mice. So the kind of practice effect idea that if you test them every day, they're going to get so good at the task that it doesn't matter how much amyloid beta deposition they have, they won't show deficits. This one. Now, to put these ages in context, you can think of, 28 to 40-week-old mouse as a young, healthy adult mouse. 65 to 70 is a pretty old mouse. And for reference, I didn't have time to show you these data, but in Elena's paper, they showed deficits in the transgenic mice at about 48 weeks of age. Let's check my time. So we analyzed our delayed match to position data using a forgetting function here where we calculated log D values and fitted them using the forgetting function displayed there. So what that function tells us is that log D values, a measure of accuracy in the task, declines as a function of the delay between the sample lever presentation and the opportunity to choose between the two levers with two parameters. A, which defines the intercept here, so the initial level of accuracy, and B, the rate of forgetting. Just to give you an idea, because the data I'm going to show you are all the parameter values, I want to make sure that you're comfortable with what those parameter values mean. So in this graph, you can see two curves, solid and dashed, that were generated using different A values and the same B values. So different A values means they start at different points on the y-axis, but they decline at the same rate, or start at the same A value, the same intercept, and decline at different rates. And so we were most interested in whether the Alzheimer's transgenic mice would start to exhibit higher rates of forgetting as they aged and the neuropathology developed. Now, before I show you the data, I want to orient you to how the data are going to be presented. So on the left, I'm going to show you the data for the continuous testing group, and on the right, the data for the intermittent testing group. And I have the graphs broken up with these vertical lines. The vertical lines indicate the points in time when the intermittent testing group went on break. Remember, the continuous testing group never took breaks, but to facilitate comparisons of equivalent points in time, I put those vertical lines on the continuous testing group graph. And then the non-transgenic mice data are shown in open symbols and transgenic mice data in filled symbols. So in terms of the parameter that defined initial discriminability, you can see in the continuous testing group was maybe a slight improvement over time. So the mice do tend to get better in the task, but no age-related decline in initial discriminability, how well they do at the short delays. The initial break that the intermittent testing mice took produced a drop in initial discriminability, but they quickly recovered to their pre-break level. So they take a break, they come back, they don't do as well in the task, but they quickly return to their pre-break performance levels. And we didn't see that same effect after the second break. In terms of the rate of forgetting, we saw over time they did get better. So this is like the practice effect that we were worried about. Both genotypes are improving. Their rates of forgetting are going down, which means that they're performing better at those long delays. But by 65 to 70 weeks of age, these continuously tested transgenic mice are showing a persistent deficit. So they have higher B values, which means they're doing more poorly on those long delays than the non-transgenic mice. In contrast, in the intermittent testing group, you can see that the break produced an increase in their rate of forgetting. So it impaired performance when they first came back, and they didn't recover that quickly. So it took them many weeks to return to sort of their I guess they went even down lower here, so they did show some improvement by the end. But one important aspect of this is it looks like the non-transgenic mice recovered more quickly, which is why we get some genotype differences here. So the non-transgenic mice recovered to pre or even better than pre-break levels more quickly than the transgenics did. But the transgenics do show recovery almost back to non-transgenic levels by the end of that testing period. And then after the second break, they again show an increase in forgetting that slowly recovers with time. Okay, so we saw age-related, delay-dependent deficits in the continuously tested transgenic mice. But it looks like breaks in testing actually increased forgetting and obscured the between genotype differences we were looking for. So it's actually opposite what we were worried about. Because the break in testing impaired performance of the non-transgenic mice, it decreased our ability to see those between genotype differences, whereas in the continuously tested mice, the non-transgenic mice just kept getting better, whereas at some point the transgenic mice stopped getting better and actually started to worsen, and it was easier to detect that difference in that group. So we still have a lot of work to do. We are going to analyze their brains and look at beta amyloid levels. We might be able to correlate individual differences in beta amyloid neuropathology with declines in performance. It seemed that deficits developed at a later age in our mice compared to Elena's data in the water maze. And there are a couple important differences between her study and our study that jump out to me at least. One is that we use positive reinforcement, and it's negative reinforcement, of course, maintaining water maze performance. Our mice are food restricted, whereas her mice were not, and we know that food restriction imparts a number of health benefits, and so that may have, that'll be our recommendation. You should eat less if you are diagnosed with Alzheimer's. But from my interest, I want to move on to intervention. So we have deficits, we can start to test things to alleviate or delay the onset of those deficits. So I've been focused on drugs to potentially treat cognitive impairment, and that's something I would like to do. But through some of my interactions with some of my collaborators, I've become interested in other potential interventions, like adding omega-3 fatty acids to the diets of these mice to see if that delays or alleviates deficit development. Okay, so how am I doing? Okay, we're good. So I just want to mention a couple other technologies that I haven't had the benefit yet to work on, but I think provide exciting opportunities for behavior analysts to collaborate with other biologists and neuroscientists. So one technology is conditional knockout technology. So as I mentioned, one problem, or one concern about the type of knockouts that I have shown you data for is that the product of the gene, be it the receptor or the signaling molecule, is eliminated for the duration of the animal's lifetime and in every tissue in the animal. Conditional knockout technology allows you to be selective about disrupting a gene. You can be selective in terms of when you knock it out and where you knock it out. So you can knock it out in a neonate up to an adult animal. You can knock it out in a particular brain region. So you want to know whether the thalamus is involved in a particular behavior or the gene product acting in the thalamus, then you can eliminate it only in that brain region. And then a couple other exciting technologies. Many of you may have heard about optogenetics. It received or is receiving a lot of media attention, probably in part because you get cool pictures like this. But what it is you can genetically engineer expression of light-sensitive receptors in distinct populations of neurons in particular brain regions. And then you can implant a light-emitting device in the mouse or rat brain, and then you can turn on or turn off those neurons by shining a light, which stimulates those particular receptors, which can be either excitatory or inhibitory. So you basically turn on or off neurons at will with this technology. And then another one that may be a little easier to work with, because that requires brain surgery and maintenance of the indwelling device, is a thing with this really long name, DREADS for short, which stands for Designer Receptors Exclusively Activated by Designer Drugs. What that means is you can get an animal with neurotransmitter receptor that has been slightly altered in such a way that it's no longer sensitive to the endogenous neurotransmitter, and it is only sensitive to a designer drug, a synthetic ligand, which is supposed to be inactive otherwise. So then, if that all works, you can now give that animal this synthetic ligand and stimulate or turn off receptors that are expressing these neurons that are expressing these designer receptors. And you can do that in the normal way we give drugs. You can put it in the food, you can IP inject them, you can put it in the drinking water. So it's a much easier technology once you have the animal to take advantage of. Okay, so to give you some final thoughts, I think that advances in biological and genetic technologies offer fruitful collaborative opportunities for behavior analysts, and I hope if any of you are interested, any graduate student possibilities out there, contact me. But I hope that behavior analysts see some possibilities in these technologies to work with folks like Chris Morrison and others. And then finally, I'd like to suggest that, or I'd like to think that although Skinner could not probably have envisioned the development of these technologies, I'd like to think that he'd be willing to work with some of these folks if he was still alive today. And I just want to acknowledge my various collaborators on these projects and thank you for your attention today. Got time for questions? Happy to answer any questions. Yes, sir.
So, I may be wrong about this, but I think there is anecdotal evidence that smokers are less likely to suffer symptoms of Alzheimer's, and so your preparation here, when you were talking about pharmacotherapies, were you talking about maybe nicotine?
No, I've been thinking about like alpha-5, GABA, negative allosteric modulators.
I don't think you smoke that.
Yeah, but that would be a lot easier. So you tell them to smoke or just get a patch. I'm not aware of that. That's interesting. I'll have to look into that.
Maybe wrong.
Yeah. Yes?
Related to that, I was just wondering if you could comment on the dietary interventions and the omega-3s, and you said you've recently gotten interested in it.
So I have a collaborator who is interested in metabolics. So all the people at Pennington do metabolism and obesity and diabetes, and some of them look at dietary interventions like omega-3 fatty acids to reduce inflammation associated with those. And I don't know these data very well, but there are some data suggesting inflammatory processes in Alzheimer's. And so at least at a very rough level, you could see like if you could add that to the diet and it reduced those inflammatory processes, maybe it would confer some benefit in Alzheimer's.